Uh, Good evening to the participants and our trainer, Dr. Rajat Nath. On behalf of Growing Seed, I would like to welcome you all to the ninth session of our 21 days long uh, workshop on biological data analysis and bioinformatics. So, in previous classes, we have learned about biological data analysis and some of our bioinformatics also. So today the class will be taken by Dr. Rajat Nath, who is currently working as a research scientist at Assam University in Life Science and Bioinformatics Department. So Dr. Nath will take classes on bioinformatics and drug designing also. So I'd like to invite Dr. Nath to kindly start the session. Dr. Nath, kindly start. Thank you.
Thank you, Poonam, and good evening, everyone. Am I audible?
Yes, Rajat, you are audible. Please start.
Okay. So, um, yes, and Poonam said, I am Dr. Rajat Nath and currently working as a research scientist in Central Laboratory of Department of Life Science and Bioinformatics, Assam University. Silchar. So, today we will learn about the computer-aided drug designing. So, what is the basis of computer-aided drug designing? Why we choose computer-aided drug designing? Besides the traditional methods, and what are the procedure or the steps followed in computer drug designing. Okay, so I will request everyone, please interact during the class so that we can learn easily, and otherwise I will talk and everyone will listen and I will not understand that everyone is understanding or not. So please interact during the class. The slide is visible or not? So why CADD? CADD means computer aided drug designing. And why not traditional drug designing? So basically, there is two most important reasons why we chose computer aided drug designing over the traditional drug designing. The first one is the cost, and the second one is the time. So to develop a drug by traditional methods, it may take 10 to 20 years. And after the completion of all the work, after 20 to 20 years, 25 years, we'll get nothing. Okay, so maybe we have identified one compounds or one drug list by traditional methods, but after doing all the things in the clinical trial, and the result shows unsatisfactory result. So that's why the cost and the time both are got lost. So in computer drug design, what can we do? We can predict that one compound is working for this particular disease or not with a very short period of time. Okay. So the first point I mentioned here is time efficiency. That means the computer drug designing allows rapid virtual screening of potential drug candidates. That means you can screen more than 1,000 compounds at a time with computer drug designing. But in traditional method, you can't. In traditional method, you can work with one compound at a time. But in case of CADD, you can screen more than 1,000 compounds at a time. It will reduce the time and also the cost. Second one is the cost effectiveness. Yes. So in traditional methods, we have to do the clinical trials, in vivo studies, in vitro studies, and the chemical and the consumables cost lakhs so to complete the experiments. But in silico drug designing, you just need a computational system which is cost around for simulation and server. It will cost around 20 to 30 lakh system. You can do lots of work, but for a single compound in traditional drug discovery, it may take crores. So here we can see that it is cost effective. High throughput skill analysis. That means vast number of compounds we can screen in a short period of time, and precision and target specificity. So when we do traditional drug designing, we can only assume that it may work on certain type of diseases, but we can't say which pathways is following by this group of compounds or the crude extract. But in case of CADD, we can predict the possible pathways by which the compound is working to treat the disease. Then, when we predict the compounds after by computer drug designing, then we go for the clinical trials. The chances of failure of experiment is very less because we already predicted and screened out of thousands of compounds to get the best ligand for a particular disease. So it reduces the experimental failure. And as I said, that we can get the molecular mechanism insight by the computational drug design. In traditional methods, it's very difficult to get the probable pathways which the compound is following. And as that this era is of big data, that is very much RNA data and some other data are very vast data is available, and by single-handedly it is very difficult to handle all those big data. But in computer drug design, we can easily handle those data sets, and we can analyze our findings by those big data. And we can do very easily collaboration with other institution by computer drug design, because we no need to send any compounds, we no need to send any biological samples to other institute to collaborate. We just need to send some data file which I can send very easily by mailing them. But in case of traditional drug designing, the sending of a biological sample is very hectic, and sometimes it degrades by heat. So it is very difficult to send one biological sample from one institute to other. So it is very easy for computer drug design to collaborate with other institutions. So this is the overview of computer drug designing. First of all, there is two kind of drug designing. One is ligand-based drug designing. One is target-based drug designing. What is ligand-based drug designing? When you have a choice of ligand, you have worked with any biological sample and you have found one compound, and you think this compound maybe act as a drug. So you have a compound, but you don't know in which disease it can work. So in this case of drug designing, we go for the ligand-based drug designing. But if you want to work with a particular disease and you don't have any drug leads, but you want to work for a particular disease, in this case, we go for the target-based drug designing. So for target-based drug designing, first of all, we need for ligand-based drug designing, we need a ligand. So the ligand may have available in databases. So we will download this ligand. But in case of if the ligand is not available in the database, we have to draw the structure. But the question is, from where we'll get the ligand? So if anyone is working on phytochemistry, so you may get this ligand from any medicinal plant. You may isolate one ligand from a medicinal plant and find that this compound having any drug-like properties. So you are interested to work on this ligand and you find a cure for some kind of ailments. So for this, you will go for the ligand-based drug designing. So you will first search that is this ligand is available in the database, because in computer drug designing, we need this ligand as .hdf format or .mol format or in SMILE format. So if this ligand is available in various database, so you can easily download this 3D structure of the ligand and you go for your work. But if ligand is novel, you find this ligand and no one was reported is earlier, so you have to draw your ligand and and need to convert in 3D format and download in HDF file, mol file or a SMILE format. So what is SMILE HDF mol? This is nothing but the uh, different uh, format of your SMILE of your ligand. So various software we use during our computer drug designing, some software accept SMILE format, some software accept HDF format, and some software accept mol format. So we need those type of formats of our ligand ready for different types of work, so that we can put or upload our ligand to the portal and can access our result. Another avenue for target-based ligand-based drug designing is combinatorial compounds library. In this process, what we do, if we have a ligand and it's showing some type of toxicity, or it is showing drug-like activity, but there is some uh, problem in this ligands that it is not able to cross the cell membrane, or it is having the solubility issue. So what can we do? We can make a library of changing that side change of your ligand and predict those structure or predicted structure, so that this compound library is showing better result than your selected compound or not. Then comes the target prediction. It is known as target fishing. So by this process, what can we do? We will upload our ligand, then it will predict the probable target protein, enzyme, or other hormones, that there is the probability that this ligand can bind with those target. So from the list of target, we can identify or we can predict that which type of activity this ligand can show, or which type of disease we can target for our further studies. So this is a very important process in ligand-based drug discovery, and it is known as target fishing. Then comes bioactivity prediction. There is another process. This is called bioactivity prediction. In this case also, we will upload our ligand and it will predict the probable bioactivity. That is it showing any anti-cancer activity or not? Is it showing any hepatoprotective activity or not? Is it showing any neuroprotective activity or not? By that, we can assume that this ligand has this type of activity. So we can focus on this type of disease and we can go for further work. Then come drug likeliness. The scientist Lipinski give a rule. This is known as Lipinski rule of 5. So we can see that this compound is following these rules or not. This technique is known as drug likeliness. Then come admetox. Admetox means absorption, distribution, metabolism, excretion, and toxicity. So to become a ligand, a good drug, it should follow all the properties of admet or admetox. If the compound is not absorbed by our bloodstream, it can't go to the target to show its activity. If the compound is not distributed in our body, then also it may have a problem that if the ligand is not distributed, it can't go to the our target of interest. Then comes the metabolism. It is very important because after the work, this ligand should metabolize and excrete out from our body. And in some cases, we deliver a big site of compound in our body, then it got metabolized in our body, and the metabolite form of this compound is shown as a bioactivity. So metabolism is very important for become a drug. And excretion, the compound should remove from our body after its activity has done. So good excretory profile should have your ligand. And toxicity is most important. We have to see that this compound is having any other toxicity or not in our body. So that also we can do by computer drug designing. Then when come about our target-based drug designing, so in target-based drug designing, we are working in a particular type of disease. Let us consider you are working with a cancer. Okay. So though you are working on cancer, so you know which target you are trying to inhibit by your drug compound. Okay. So in by your research or by literature study, you will find the target which you know if you inhibit it, then it results the cure of cancer. So where from where you can get this target? This target you can find by literature or by your uh, you design of your org. There is another process. This is known as gene network analysis. It is also a in silico process by which you can study all the genes related to that particular type of cancer. From this analysis, you can identify the most crucial gene which is responsible for progression of cancer. By this process, you can get a gene of interest, and if you inhibit this gene, it may cure the disease. So this is another avenue of your computer drug designing or bioinformatics. This is known as gene network analysis, by which you can identify the gene of interest for a particular disease. It may be a novel gene you will uh, identify by this gene network analysis, which may not reported earlier. So it may have a good chance of uh, avenue for drug discovery, that you have reported one new gene target or new target for a disease, and if it work, then it may change all the scenario for the particular type of diseases. And again, if the target protein is available in the databases, then you can download this in pdb format. And if this target protein is not available in the databases, then you have to predict the structure. And this is called homology modeling. So what is basically this prediction of the structure? Because the target is mainly protein. We all the targets for we target for a particular disease to cure a particular disease. The target is normally a protein structure. So we get the protein structure from the PDB or UDPROT. This is the two database, famous database for protein. So from where we can download all the protein structure. But in some cases, if the protein is novel, as I mentioned, that if you got the protein from gene network analysis and it is novel, nobody has reported it earlier. So definitely it will not available in the databases. For this case, you have to predict this protein structure, and this is called homology modeling. And it is also an in silico process. Then when we got our target and we got our ligand, we'll go for the protein ligand interaction studies. For that, we do the molecular docking analysis. What is molecular docking analysis? It is nothing but we see the interaction between the target and the ligand. What is the target? Target is a protein. And what is ligand? Is my drug candidate. Okay. So where this drug candidate get bound to the protein, in the active site of the protein. So in molecular docking, what we do? First of all, we will determine the active site of the protein. Then we go for the molecular protein ligand interaction. So in case of we know in chemistry this ionic bond is the strongest bond, but in case of biochemistry the hydrogen bond or covalent bond is the strongest bond. So if the number of hydrogen bond is forming ligand with the target, the stronger the interaction will be there. So We know when a ligand is bind to a target spontaneously. That means when we send this ligand to bind with a target and it got bind in that case, what will happen? The energy will release. When the ligand and target bind spontaneously, the energy got released. That's why when we got the molecular docking score, there will be a negative sign. That means this ligand is bind to that target and this much of energy is released. The strongest the bond, the higher the energy will release. So that means negative sign will be there because this is means that energy is released and the value of the score is higher means the bond is the stronger. But in case if the ligand is not get uh, attached to the target spontaneously, in that case we have to give some energy from external so that it get bound. So in that case the molecular docking score will be in positive. But in our case we don't give the energy from external, we need the spontaneous binding, so we always consider the negative score of the molecular docking. Okay, so what is molecular docking we have understand, but in case of molecular docking what happened, what is the limitation of molecular docking? In molecular docking the protein structure will be in stable condition and the ligand also will be in stable condition, it will go and bind. But in the living system or biological system the protein is not like that and the ligand is also not like that. In living system there is various factor which is affect their binding. There may be a pH, there will be water, there will be lipids, there is responsible temperature, there will be ions, there will be salt concentration, and the protein structure is not in rigid format. It will continuously try to change its conformation and ligand also changing its conformation. So this type of interaction we can't see in molecular docking. That's why we go for the molecular dynamic simulation. In molecular dynamic simulation, we can mimic the structure of protein, the change of conformation of protein, the external factor we can give in a box that is called the molecular dynamic simulation box. We give the all the responsible properties available in our biological system in this box. We'll insert this protein in this box, we'll insert our ligand in this box, and we see how this is interacting. Okay, so after molecular docking, when we've seen that it is working fine, then we go for the molecular dynamic simulation where we simulate the biological process already going on in our living system. We mimic the process in molecular dynamic simulation. It is also a computer aided drug designing process. Then we go for the QSAR. QSAR is the qualitative structure analysis relationship. In this case, we can predict the bioactivity of this ligand. In that case, what we'll do, we give a data set to a particular to the software. Like that, if we consider a boy in a class, is he is a good or bad? So if you don't have any idea what a good boy means or what a bad boy means, you can't say that is this boy is good or not. So you have to know what character should have to become a good boy and what character should have be in a boy to become a bad boy. So like that, we give some descriptors, molecular descriptor to the software that if the descriptor is like that, the bioactivity will be like that. It is known as training data set. So we provide the training data set to the software and we'll provide our characteristics or descriptor of the our ligand, and that software will interact all with all the training data set and give us the predictive bioactivity result. Okay, so that's why we like that we do the QSA. So this is the brief overview of computer drug designing. What we do to complete a drug designing study from starting to ending. Okay. After getting the result from molecular dynamic simulation and QSL that yes, this compound is working against this disease, or yes, this disease is inhibited by that particular compound, we go for the in vitro studies or in vivo studies or clinical trial. So we by this process we save lots of time and lots of money by this process, and we can screen thousands of compounds at very short period of time. So after completing this completed drug designing, when you go for the in vitro or in vivo studies, the chances of failure or chances of the failure of the experiment is very low. So this is why we chose, and modern era is working on computer drug designing more than the traditional drug designing. Okay, so this is was the overview of computer drug designing. If anyone having any confusion during this much of information, please interact. If anyone having any doubt till now, today we'll, today we'll do the ligand based drug designing. Okay, so before going to start the training, if anyone having any doubt or confusion, you may ask. Okay, so MS or HPLC data, can we go for identifying the compounds if it has only any? Yes, yes, yes. Uh, when we work with the computer drug designing in phytochemistry, we go for the GCMS or LCM, HPLC. In HPLC, you will not get the compound details. You will go for LCMS. Okay, you go for LCMS, then you will get the available compounds in your sample. Then we can go for the antimicrobial activity prediction. You can do it by two way. Okay, you can do it by target by ligand based drug designing also, by in target based drug designing also. If you know the target for antimicrobial properties, that if this protein can be inhibited by those compound or not, in this case you just download the 3D structure of your protein of interest and go for the molecular docking and see these compounds are inhibiting this compound or not, this target or not. And if you go for the ligand based drug designing, you go for first you go for the drug likeness properties, admit properties, and bioactivity prediction. Then screen out the compounds which is not showing the drug like properties. Then the shortlisted compound go for the target prediction and see is their target for antimicrobial properties is available or not. If there is any target which is having the antimicrobial property, you go for this target and go for molecular docking simulation and so on. So you can do. So if we go for the ligand based drug designing, first of all we need a ligand. As Abhishek said that by HPLC or GCMS, he may get a ligand or drug-like compounds. Okay, so we assume that we get a drug-like ligand and we will start working with this ligand. So what we'll do first of all, just let me share the screen. So just consider that I have got a compound or ligand from GCMS. So first of all, I have to check this ligand is available in the database or not. The most famous database for this ligand is PubChem. Just type PubChem. This is maintained by the National Institute of Health. So this is the PubChem database. So what you have to do, that if you got your ligand name, just search your ligand name. Let us consider that I have found a ligand from GCMS or LCMS is known as just like aspirin. This is the compound I have found by GCMS analysis. Okay, when I search this aspirin, you can get so many results. This is 107 compound result is available. So just check the best match and see the name is exactly what is typing or not. So this is aspirin, what I am looking for. So just click this one. This is the aspirin. This is the structure. This is the 2D structure of aspirin. Now just scroll down. See, there is known as written as canonical smile. So as I mentioned earlier, that we need HDF format, mol format, or smile format to conduct any biological experiment in computer drug design. So this is the smile format. This is the smile format we are looking for. This will require for various bioinformatics analysis. This is the one process from where we can get the canonical smile. Then we need HDF file for our experiment. Just in right hand side, see there is written as download. Just click on download and you will get 2D structure and 3D conformer. So we never select a 2D structure, because if you select a 2D structure, it will not get the perfect or actual result during your bioinformatics analysis. It should in 3D conformer, because if structure is 2D, it will not give the accurate result. It should be in its 3D conformer. So see, there is four data types are there. One is HDF, there second one is Johnson, third one is XML, and fourth one is ASNT. So we generally need HDF format. So just click save button below the 3D conformer in the HDF format. So when I click the save button, there will be a name. I will writing the compound name as aspirin. Okay. I have downloaded the structure. Now you see here is written as PubChem CID. This is 2244. That means this aspirin is identified by PubChem by a ID. It is 2244. So as we downloaded this structure, I have written the name aspirin, but it is not aspirin. When we will open this structure in any software, just I am opening a docking software. This is a one docking software. I am importing this compound to this docking software. What you will get, this is one ligand it's showing. Now, see, the name is written as 2244, as same as written in the PubChem ID 2244. So if you upload 100 of ligand and it is written as this type of format, we can't understand which compound we are dealing for. If the second compound is writing as 1, 2, 3, 4, third compound is 5, 4, 6, 1, then we don't understand, we will not understand which compound we are looking for. In that case, I have to do one thing so that it show its name as aspirin. For that, nothing have, just right click this structure and go for open with notepad. So this is the HDF format of my structure. In the top most line, you can see there is written 2244. You just repeat this word with the chemical name. But remember, if your chemical name have any space, you should avoid this space. If there is name one compound name is gallic acid. In that case, what we do? Gallic and then given one space. But you can't give space in this format. What you can do? You can just give an underscore if there is a space in your compound name. Okay. If it is one word compound, then there is no problem. I am writing this name as aspirin and click Control S. Just click Control S and close the. Now I am inserting this compound to the docking software. See, now it is showing its original name, aspirin. So if we rename the compound, then you can do your analysis very easily. Otherwise, it will be very chaotic if you don't rename your compound. So I am closing this docking software because it will not require now. So now we have this compound HDF format. Now I am this is the 2D structure of your compound of interest. Okay. So if your compound is not available in the database, what you can do? In that case, this structure you will get through the NMR or crystallography, you will get your structure, predicted structure. So you have to got the chemical canonical file of structure for your bioinformatics analysis or HDF file of your structure for bioinformatics analysis, but it is not available in the database. In that case, you can draw this structure in various drawing software. So two software I will recommend you should download. It is one is Cam Sketch and another is Cam Draw. Just write down the name. One is Cam Sketch, another is Cam Draw. Cam Draw is a package comes with the Cam Office. Okay, so Cam Sketch you can download easily. It is freely available, but Cam Office is not freely available. Okay, so you can search in Google. You can get a cracked version of Cam Draw. So let us consider you don't have the structure in your database and you have to prepare your structure. So I am opening the CamDraw software. CamDraw. You just download in your system. If anyone having trouble with downloading, just let me know. I will provide the software. So this is the CamDraw software. I am taking a screenshot of this compound so that I can easily draw it by seeing the structure. I am pasting it here. Okay. So I have to draw this structure. See, first of all, there is a benzene ring. Okay. So in left hand side, this is the toolbar. You find the benzene ring. This is the benzene ring. Just click on the benzene ring and click on the white space. So this benzene ring I have drawn. Now I have to give the double bond in alternate. So in left hand side, see this is the structure of double single bond. I have already one single bond, so I need another single bond here. So if I click outside, it will like show like that. So it will not be connected with my structure. So if you mistakenly click outside the benzene ring, you can erase this structure by there is a eraser tool. Just click on the eraser tool and erase the structure. Okay. So I am again selecting this single bond and just go to this benzene thing. See, when I go to that benzene ring, there is showing a blue color, blue highlighted color. If I get this blue highlighted color in this bond, and I click, there will be a double bond in this single bond. If I click in the corner, it will be a single bond outside the benzene ring. So it is, I not require this one. What I will do, I will click the eraser and erase this structure. So I will draw all the double bonds. It is, the benzene ring is completed. Now I need a single bond upwards to the benzene ring. For that, I will point out to this point. There is showing a blue line. Just click and just move this cursor like this by clicking the left key. So when it is the upwards, that means the required structure is able. Now I need another single bond in right side. So just click. If I click in the middle of this single bond, it will give a double bond here. And see, there is a red mark. That means there is a carbon, and carbon may have four bonding. But in this case, there is one bond, two bond, three bond, four bond, and five bonds. And carbon can't make five bonds. So there is showing a red mark. When I will delete this extra bond, it will show no warning. Okay. So like that, we can create our bonding. So I need a bond in from this single bond. So I have to click on this blue dot and click and move. See, it is moving. So I need a bond like that. Another bond I need in that side. Another I need there to double bond. Then there will be oxygen, and oxygen will be connected with a hydrogen. So I will add oxygen here. For now, I am making the bond for hydrogen. Then I need another bond in that part. There will be oxygen. There will be another bond. There will be another bond, and another bond. So this is the, then I can select this A, and wherever you need to add the hydrogen or oxygen, just go to there, and when blue highlights is showing, just click and write the letter like that. There is a oxygen, there is a hydrogen, there is another oxygen, there is another oxygen, and there is another oxygen. So see, the structure is completed. So I am showing the structure by another way. See, I am this structure is drawing by another methods. There is a benzene ring. This is the benzene ring, and you can see that this is a half benzene ring. If I consider this part, there is a half benzene ring. So I am drawing another benzene ring over this arm, and in that side also there is a half benzene ring. So I am also drawing another benzene ring in that side. And if I see that there is another half benzene ring, so I will draw another benzene ring in that side. Now I will take the eraser and erase the unnecessary bonding. This bond is not required, this bond is not required, this bond is not required. In that case, this bond is not required, this bond is not required. And in that case, that much, this bond and this bond is not required. Now I have double bond here, double bond here, double bond here. I will put the oxygen, oxygen, oxygen. In that case, in this area, I have written this oxygen bonded with hydrogen, but you can write OH at a time. It will not change any chemical properties. After completing the drawing, just select this arrow, and when you select this, it will select the structure. Okay. So This is one method I have drawn. This is by another method it is drawn. Oh, sorry, there will be another single point. Okay. Now it is perfect. So I am deleting this part. You can draw by any methods. However, you can. I am deleting this also. Now select this selection button, and this is your structure ready. Now there is another method you can draw your structure. If you know the IUPAC name of your structure, you can easily generate your structure by the name. So I am going to the pattern again, and below you can get the IUPAC name of your structure. See, there is the IUPAC name of your structure. So just select this name, go to camp draw, and go to structure, then convert name to structure. Okay, structure, convert name to structure, and paste the IUPAC name and click OK. See, the structure is generated. It is rotated 90 degree. If you rotate clockwise 90 degree, it will be same as the structure I have drawn. So if you know the IUPAC name, you can generate your structure very easily in ChemDraw. So there is two methods by which you can generate your structure. So now I need the smile format of this structure. For that, what can I do? I will select this structure, copy it, and I will open another software. This is CamSketch. So this is another software. This is known as CamSketch. In CamSketch, we just paste our, just Control V to paste your structure here. This is the structure I have pasted. Go to the tools, then go to the generate, and go for the smile notation. See, this is your smile. Smile notation of your compound. Sorry, ligand. So this is the smile notation. You can write the IUPAC name here also. Name for the structure. See, it has given the IUPAC name also. So another thing you can do here in this software is we have canonical smile, and if we copy this canonical smile and go to the tools, then generate, then structure for smile, and paste the smile and click OK. See, it will draw the structure for us. So in this software, we can get structure from smile, name from structure, or structure from the smile format. So this is the smile format we need for our bioinformatics analysis. Now, this is the structure. This is the smile format. But if we need HDF, what can we do? For this, there is another software known as OpenBabel. OpenBabel you can download easily. It is freely available. So just download and install in your computer. So this is the OpenBabel. In OpenBabel, in left hand side, input format, and right hand side, output format. So I will input the smile format. So just click on the smiles, then click on input below. So what I can do, I will copy this smile, go to OpenBabel, paste it, give a space, and write down the font name. But remember, there should not be any space. Okay. So if there is two word name, you should use a underscore or dot. Then go for the output format. I need HDF. So just scroll down and found the HDF. I have find the HDF. Then select the output file. Just click on the three dots and name the I'm writing this name as aspirin 2 because aspirin is already having in my system. So I'm writing as aspirin 2. Click save. And most important, click on generate 3D coordinates. And below, must you create click the generate 3D coordinates and click on convert. It will take few seconds and it will convert to HDF. If we close this, see, there is a ligand known as aspirin 2 is in HDF format. So now we can see how we can retrieve a compound from the databases, or if the compound is not available in the databases, how we can draw this structure, how can generate its name, how can we generate its smile, and how we can convert this smile format to HDF format. In this software, you can do anything from the just select the input format and just select the output format. You can convert any format to any format. Okay. So this is very important software for bioinformatics. Just Google the name Open Babel and you can download freely. So we learn how the retrieval of the target ligand is done. Anyone having any query or doubt, please let me know. Sir, why 3D structures are selected for molecular docking? Why 2D structure give variation in docking score? Okay, for the first question, let us consider. Uh, okay, this is a mobile phone. Okay, and this is a pen. Pen structure is okay. So see, this is your ligand structure. Okay, this is a 3D structure of your ligand. I think you are able to see. Okay, this is the 3D structure of ligand. If I see right this is one line, and there is upwards line. So this is the 3D structure of your ligand. And if I consider it is a 2D structure, then how it will be? It will be like that in single plane. If I draw this structure in 2D, it will be in single plane. So this is your protein. Okay, let us consider this is your protein, and this protein is going and binding like that. Okay, this is a one plane, and this is also one plane, and get binded like that. But your protein ligand is not like that. Now your ligand is actually like that in 3D format. In 3D format, like that. So can it go and bind like previous one? Because it will inhibit these two arms to get attached with the target. Okay, it can't go and attach with this target. But in case if it chooses 2D structure, it easily can get and bind with the target. Okay, so that's why if we chose the 2D structure, which is not the actual structure of the protein, or sorry, ligand, it is just for showing or representing a 2D plane or 2D display, we showing this structure as like that. But actually, it is not like that. That's why 3D structure should be there for molecular docking analysis. Are you clear? Okay. And can you repeat one more from the smile? How can we draw static? Okay. So just go to the, let me share the screen. Okay, just go to the cam sketch, deleting all the things. Just go to the cam sketch, go to tools, and go to generate. In generate, there will be a structure from smile. There is an option called structure from smile. Just click it. Then paste your smile. This is your smile. And click on OK. Then it will be, then click anywhere, it will paste your structure. Okay. So this structure is looking not exactly looking the like the previous structure. For that, what can you do? You can go for tools and go to clean structure. Just click on the clean structure. See, that structure has been changed like previous one. You can draw your structure in this software also, but it is very difficult to draw any structure in this software. That's why I recommend to draw the structure in CamDraw and do all the other necessary analysis in Cam Sketch. You can get the chemical description of this compound by this structure also. Just go to tools, go to calculate, then go to all properties. Tools, calculate all properties, then copy to editor. See, all the chemical nature of this ligand is calculated, like formula weight, molecular formula, composition, molar reflectivity, molar volume, paracore, everything is calculated. So this is the chem sketch. So anyone having any doubt or go for the further analysis? Okay. So now we will see the drug-like likeness of our compound. Okay. Is it having the drug-like properties or not? First of all, after getting our compound, it should act like a drug. So if it is not looking act like a drug, then there is no reason to go further with this compound. So for this, there is a server known as MolSoft. Just type MOLSOFT. Click MOLSOFT. MolSoft. Then scroll down and just click this drug likeness. Click on this drug likeness. So this is the drug likeness molecular property prediction of MolSoft. So this is one compound structure already they have given. We have to erase the structure. There is an option clear. See, there is an option. It is fifth option. Just click this clear option. Okay. You can draw this your structure here also like that. As we have draw in our cam draw, we can draw there. But no need to draw there. Just in sixth tool, there is known as import. Just click this one and paste the smile font. So where is our smiley? Uh, just paste the smile. This is our smile. Just paste. So I have pasted my smile and click OK. See the structure already drawn. then go to the calculate properties. Now you will get some data here. So there is a popular formula given by the Lipinski, known as Lipinski rule of 5. There is 4 rules, but it is known as rule of 5 because all the properties he had mentioned is either 5 or the multiplication of 5. That's why it is known as rule of 5. Yes, but it's there is 4 rules. Now, what is the rules? To be a good drug, the molecular weight of the ligand should less than 500. The molecular weight should be less than 500. Number of hydrogen bond acceptor, HBA means hydrogen bond acceptor, should be less than 10. Number of hydrogen bond donor should be less than 5. And log P value should be less than 5. So there is 4 formula and known as rule of 5. One is molecular weight should be less than 500. Hydrogen bond acceptor should be less than 10, and number of hydrogen bonds should be less than 5, and log P value should be less than 5. So see, the molecular weight is showing 180, number of hydrogen bond etc showing 4, number of hydrogen donor is 1, and log P value is 1.122. So all the rules of Lipinski is following by this compound. Okay. If any rule is violated by this compound, it will be written as red color. If you giving another compound here which has violated the Lipinski rule, it will show in red color. Yes, there is no violation, so all are in black. So from this we can consider that yes, this compound is following the Lipinski rule of 5 and it is having the drug-like properties. So in below, there is a score. The score is 0.27 and score is known as drug likeliness model score. The score should be nearer to 1. The score 1 is the best drug-like properties and and it should be nearer to 1. Now, why it should be nearer to 1? See, there is 3 lines. One is blue, one is green, and one is red. Blue line means drugs, green line is non-drugs, and red line is your compound. So this is the blue line. This is the peak where the drug likeness properties is highest. So if I consider this peak, you can get a value 1 because there is 0, there is 2, there is 4. So in that middle portion will be 1. So 1 means the highest predictivity of drug likeness or a drug like compound. So the value should be between 0 to 2 and nearer to 1 is the most drug like properties. So this is the peak and this is the 1 and my compound is showing 0.27. It is nearer to 1. That's why I can consider that yes, it is showing drug like properties. And if I go further there, this my compound is present there and it is showing good drug like properties. If it is in this side, that will show the non-drug like properties. So we can't consider if this red line is below the 0. Okay, so you can just copy those properties and you can take a screenshot of this graph and you can proceed this analysis to the manuscript or for your PhD thesis or anywhere where you can submit by just take a screenshot of this graph and take all the necessary properties and you can write accordingly. So this was the drug likeness. Then I will go for the target fishing. As I got that, yes, this compound is having drug-like properties, so I have to understand which type of disease it can cure. Okay, for that, one popular server is there that is known as Swiss target prediction. Swiss target prediction. So just click this server is maintained by the Swiss Institute of Bioinformatics, Switzerland. So here I have shown the Swiss target prediction. See, now there is species. There is Homo sapiens, Mus musculus, and Ratus. So if we considering to cure any disease, we are genuinely working on Homo sapiens or for human treatment to human. So the target should be of human. So we'll select the species as Homo sapiens because we are creating this drug for the illness happening to Homo sapiens. So I will select the species as Homo sapiens. Then again, you can draw the structure here, but I will paste the smile. There is the option paste a smile. So I will paste the smile. When I paste the smile, see the structure has been generated. And I will click this predict targets. It will take few minutes to predict the targets. So these are the target class, which type of target have been predicted by this software. The color denotes, what is the color denotes is written in here. That is like for blue, it is oxidoreductase. For this pink, it is a electrochemical transporter. For green, it is lyase. Like this is the chemical class. Now, I am going in left hand side. You can see show 15 entries. Just click here and click all. Okay, now you will show all the targets predicted. Now you can see there is a probability and there is a box of green color. Okay, the the green and color filled by this box. If it is fully green, that means there is probability of 100% of target this protein. In that case, there is no green color is in this box. That means there is very less chance that this target will bind with this protein. Okay, so we will not consider those targets or protein which don't have any probability denoted by the green color. So we will consider only the proteins which is denoted by the green color of probability. So see, I can consider like like up to dual species protein kinase CLK, that much. Okay. Now see, first one is cyclooxygenase 1, second one is cyclooxygenase 2, third one is dopamine transporter. Now it is showing that dopamine X transporter is inhibited by the compound. So if you are working on neuroscience, then you may know that dopamine transporter is related for Alzheimer's disease or Parkinson's disease. So you may have an idea that okay, it is having effect in dopamine transporter. So this compound may have any effect in neurodegenerative diseases. So you can go for the neurodegenerative diseases and you can further work on it. Then there is so many target. There is another target DNA lysis. So these DNA lysis may be effect in any DNA replication or anything. So this is related to DNA replication like cancer, etc. You may consider this one and go for the further studies. Estrogen receptor beta. The estrogen receptor beta is associated with some antibiotic or what, you can go for this type of diseases. Then DNA excision repair protein, that is related to cancer, because if the DNA excision repair is not working properly, it may cause the mutation. So this may related to the cancer. So when you are working with a particular disease, you will know that is this protein is related to your targeted disease or not. By this, you can consider those type of targets and go for further analysis. You can download this result by just click this. There is a Excel format, export result in Excel. Number 3. When you click it, it will save as in Excel file. See, this is the result. You can export to a Excel file. At the probability. 0.75 means 75%. 0.08 means 8%. You can just given formula is equal to into 100, and that will be the in percentage format. How much probability having this is showing the 0% probability. So we will not consider, we'll consider the upwards one. So this was the target prediction, and you can get the target by this. Another software for target prediction is Farm Mapper. This is the Farm Mapper. So in this case, as Swiss target prediction give the result within a minute, but in Farm Mapper it may take 2 to 3 days. Okay, so this is the Farm Mapper. Go to the submit job, give your file in SDF format. In that case, you have to browse and you have to upload the SDF file of your compound. This is the SDF file. Then give your email address and name a job description and submit. Okay. See, the farmer is under heavy pressure. The estimated waiting time may be longer than 14 days. That means one result you may get after 14 days. So it is very time taking, but the accuracy of the result is much more higher in farm paper than the Swiss target prediction. Swiss target prediction just give the result within a minute, but in farm paper you get the result after 14 days or 15 days, but the accuracy of the result is more higher than the Swiss target prediction. So if you have time in your hand, you can go for the farm paper, and if you have not time, then you can go for the Swiss target prediction. Okay. Okay. For agriculture, I think it will be related to plant target. Okay. So, as Swiss target prediction is only focused on the three species, that is Homo sapiens, rat, and mouse. But in farm paper, you will get the target for all the living organisms. So, if you do the work in farm paper, you may get the target of your plants. Okay, so you can go for the farm paper. It will definitely give the targets of your plant of interest. If you are having any target or predicted any target of your plant of interest. Any other queries till now? Today we will see another analysis, that is we'll see the toxicity, then we'll conclude today's class. Tomorrow we'll continue. So going for the toxicity prediction, if anyone having any doubt till now, you can interact please. Okay, so I'm going for the toxicity prediction. So for toxicity prediction, there is a server, the famous server is known as Protox2. Protox. Just type Protox. There is the Protox2 server. Just type Protox2, we will get the Protox2. Okay. So in this server, we can predict the toxicity. But before going to the toxicity prediction, you must know the toxicity class. There are 6 toxicity class denoted by this server. The class 1 is fatal if solid. The LD50 below is equal to or greater than 5. Class 2 is fatal if solid. It is LD50 below is more than 5 but below 50 or equal to 50. Class 3 is toxic if solid. It is LD50 is value is higher than 50 but equal to or lower than 300. Class 4 is harmful if solid. The LD50 value is 300 higher than 300 but lower than 2000. Class 5 may be harmful if solid. It is LD50 value is greater than 2000 but less than 5000. And class 6 is non-toxic if the LD50 value is higher than 5000. So this is the 6th toxicity class. So we go for the toxicity prediction. So going for the toxicity prediction, just click tox prediction. It is second tool. Okay. Here you can also draw the structure. You can give the PubChem name, or you can paste your smile. So I will paste my smile here. Click smile. Then your structure will be drawn here. This is my structure. Then I will choose which type of organ toxicity I have to analyze. So I will choose for the hepatotoxicity, cardiotoxicity, neurotoxicity, nephrotoxicity, respiratory toxicity, carcinogenicity, immunotoxicity, mutagenicity, cytotoxicity, blood-brain barrier clinical toxicity. Ecotoxicity I am not considering because it is not related to human health. Then I will start prediction. You can select all if you have any interest of that type of toxicity, like that acetylcholine toxicity, if you are having the only interest in organ toxicity and toxicity endpoints, you can select those. So depending upon your requirement, you can select and click on start tox prediction. So in this case, the predicted LD50 value is 250 mg per kg. Okay. So what is LD50 value? I hope you know what is LD50 value, but if you don't know, I am saying that LD50 value is means if 250 mg per kg dose is given to 100 individuals, 50% of the individual will be died. So this is the LD50 value, little dose 50. That means the amount of dose will kill 50% of the population is denoted by the LD50 value. And this compound is treated in toxicity class 3. Okay. So below you can see hepatotoxicity is showing inactive. That means this compound is not showing any hepatotoxicity. It is also showing non-neurotoxicity. It is not showing any neurotoxicity, but this compound is showing nephrotoxicity. Its probability is 69%. That is chance of 69% that this compound having nephrotoxicity. Respiratory toxicity is not showing by the compound. Cardiotoxicity also not showing. Carcinogenicity also not showing. Immunotoxicity is also not showing. Mutagenicity also not showing. Cytotoxicity is also not showing. It will cross the blood-brain barrier. There is chances of 85% that this compound will cross the blood-brain barrier. And clinical toxicity also there are having chances of 73% that they have clinical toxicity. So if your dose is 250 mg per kg or more, there have chances of nephrotoxicity and blood-brain barrier toxicity, clinical toxicity. So now you have to further go for the further studies and you have to limit the dose to overcome those toxicity of your compound. You can't go beyond this particular doses with your compound. So first of all, you see that for your cure your disease, how much dose is required, and this dose is really having any nephrotoxicity, showing any nephrotoxicity or not. So by that you can predict that if your compound having any toxicity or not. Okay, so this is just one prediction of toxicity by your compound. If all the uh, target classification is showing the toxicity, then you can go for this compound for drug because all the organs is showing toxicity. Okay, so like that you can go for the toxicity studies. So for today, we learned how to draw the structure, how to retrieve the structure, then target fishing, drug likeness analysis, and toxicity. Tomorrow we'll show the other part of this computer drug designing. Then we'll go for the homology modeling of the protein, and then go for the molecular docking. Okay, so in today's class, if you are having any doubt, query, and if you have to repeat anything, please let me know so that tomorrow I start from here. Please interact if
Okay, but I think you are done with the session.
okay. So thank you everyone. Good night. We'll see you tomorrow again, same time.
Yeah, thank you, Rajat, for this informative session. And uh, if there is any query, you may also put to the WhatsApp group so that he can uh, address it to the next session.
Yeah.
Okay, and thank you to the participants for joining. And now we are closing the session. Thank you once again, Rajat, for this session.
Thank you.
Thank you.